welcome Joao to this LCN Lunchtime seminar series. So it's my great pleasure to welcome you. And so for those who don't know, Joao is a professor of soft matter engineering in the Department of Chemical Engineering, and he's been part of the LCN for many years and associated with Imperial and the LCN for many years, starting with a PhD in polymer science in 98 to 2002, which are the same dates as me, actually, exactly. But he's moved around as well, originally from Lisbon, spent some time in Sweden and France, and as well at the National Institute of Standards and Technology in the US. But since then, mostly at Imperial with a stint also at the University of Chicago. So thanks very much. I look forward to your talk on precision polymer encapsulation in microfluidics from molecular to macroscopic scales. Thank you.
That's great, Sandrine. Thanks so much. Guys, I think there's not so many of us over lunchtime, so I would like us to have a more informal conversation, and so I drop you here my email address as well if you'd like to catch up later on after the meeting. And Sandrine and Avisa had suggested I spoke at this meeting, and I thought of sort of giving an old talk or something, and instead I decided instead that I would look for what are we actually working on right now and what are we most interested in, and we might want to engage with other people within the LCN. So that's in encapsulation. So that's taking some kind of material, valuable material, and enclose it within a capsule. And perhaps I think what we do differently in my group in chemical engineering is that we do really span the scale. So we go from the molecular scales all the way to some kind of performance, to some kind of application. This work is predominantly funded by industry, actually. So Procter & Gamble has sponsored the Royal Academy of Engineering chair, and BP and Castrol have been good partners for the development of sort of complex fluids for where the rheology is important. So perhaps just for background, encapsulation and the formation of capsules at an industrial scale for pharmaceuticals, for foods, for example, for personal care and detergents, etc., is primarily done by spray drying. This is a technique familiar to many of you. You'll take a liquid feed, you'll put it through a nozzle at very high velocities and pressures, through some kind of drying chamber that has either a co-flowing or counter-flowing drying gas. So you take a liquid, it's a bit like your deodorant spray, if you wish. You spray that into a huge tower, normally a couple of stories high, and then you will collect some dried particles in a cyclone at the end, and you probably recycle the gas back into the liquid state. You'll create particles that are normally not as nice as these ones. Often they are quite ugly looking and really just precipitated material in multiple states. Sometimes phase transitions are induced within this process, or stratifications or crystallization processes. And you'll collect some kind of mass, which by its virtue of the scale of this process, it's very important, but which we would like to control much more precisely. And so this can be done at a relatively small scale, and here are just some examples from the literature of kind of fancy looking particles where you can definitely impart some structure, even though this process is so kind of uncontrolled. So what we set out to do, my group is experimental, so we set out to work on experimental approaches to allow us to monitor kind of the sequence of steps that goes from the liquid state all the way to the formation of a particle. And we tried to think whether we could use the knowledge we have from the thermodynamics of these systems in creating functional particles. And so here's an experimental realization of this. It's a polymer that is in solution, and you can tell that the particle is shrinking. So initially the size was about here, and it's now becoming smaller. And it's revolving a bit like a globe, and you can tell that stuff is happening inside. There's a moment in which the refractive index gets matched with the matrix, and then you form a particle that is quite compact. So you can kind of wonder, you could say, well, and surprisingly, a liquid droplet is spherical to minimize the surface area and therefore surface energy. And as it dries, it creates some kind of solid particle that is also spherical. But quickly you realize that that need not be the case. For example, here you have a particle that decided to shrink and create some kind of crumpled sheet of paper instead. And you understand that quickly as well. You realize, okay, fine, that I started off with a droplet, so it's a liquid and therefore a sphere if I don't apply any external force to it. But very quickly it becomes a soft solid and indeed a solid. And so the rules of energy minimization start becoming different. And it might decide to minimize some kind of bending energy and therefore crumple instead of continue to be a sphere. So how can we study this? The droplets, as they fall from a spray tower, they go at speeds that can be many tens of centimeters per second. So that's well too fast for us to track with some kind of, for a single particle, that is. It can be done stroboscopically, of course, for distinct particles, but for the same particle, it's very difficult to track that individually. So what we decided is to turn the problem on its head and somehow levitate them. So acoustic levitation is one of the ways you can do that. It turns out to be quite simple to implement that in practice. You create some kind of dome packed with tiny loudspeakers that will apply a force that is opposed to the gravitational force. And you suspend particles very readily, and you can follow them optically, for example, but also with scattering techniques, which is what we use. And here's a particle. Typically, these are formed within sort of a couple of hundreds of microns to millimeter scale, for example. And very quickly, you realize that interesting things happens too. I mean, under the effect of the acoustic field and the opposite gravitational field, you can see that well, you can definitely arrest the particles and image them around their center of mass. You can see that they oscillate around that that node of the acoustic field. But you also might have noticed that they clearly undulate at the surface. So that was somewhere between the kind of circular profile to the sort of crumpled sheet profile. Indeed, you can imagine that if you create a skin at an intermediate condition, that skin, as the particle needs to shrink further, the skin should undulate. And so there's a buckling process that takes place. What you might have noticed as well is that as these particles dry, there were some features here that emerged on the surface. You can see these were actually tiny droplets. And again, it's not necessarily surprising that there might be some kind of condensation happening at the surface or some kind of drying. I mean, let's see that in a second. And again, unsurprisingly, here taking a model polymer, just polystyrene, you see that the sizes sort of broadly corresponding to the size of the contents within the liquid. So, but not quite linearly, of course, because as we realize, these particles are anything but compact. If you inspect them now with the SEM on the surfaces, you realize that they are actually fascinating. And so, for example, you can see that very nicely. We can excite some kind of patterns that are extremely regular at the surface beyond this kind of overall sultana shape sort of envelope of the particle. These, you can analyze them with standard methods. This is called a Voronoi analysis that looks for the number of nearest neighbors and concludes here that these types of particles have predominantly 6 whenever you excite these kind of regular shaped arrays. And these are not even the most regular that one can access. These are called breadth figures. And it's the kind of thing that you would obtain whenever you have a liquid that is evaporating, and upon this kind of evaporative cooling, you can get the deposition of water droplets, and in a nutshell, this is what you get: the evaporation of water droplets onto a skin that is becoming solid creates some kind of interesting convection currents that result primarily in this kind of hexagonal array. Okay, so you realize that there's quite a lot of interesting science to be done with these kinds of liquids that turn into particles, but that the challenge for us is to, one, be able to resolve this experimentally, and two, be able to connect the molecular scales to the macroscopic scales. So the second question perhaps is, does it matter? And of course the answer is yes. I mean, you take here this solution, which is often a property we're interested in terms of the function of these particles, and you quickly realize that depending on what cards you play, the timescales for dissolution can vary by orders of magnitude. And if you think of this as, say, a pharmaceutical drug, you might realize quite quickly that this is of interest to be able to design with the external properties. Here we're just varying the surface texture, but keeping everything else constant, that you can design the function, for example, or the dissolution profile of a tablet that could be of interest. It turns out, for a number of reasons, that you can resolve the same process instead of in air, immersing the fluid within another liquid, and we do that in microfluidics just for convenience. This allows us to tune the timescale of this sort of catwalk of processes, and also allows us to study these the thermodynamics in a more controlled way. So this process is well known to many of you. You take two inlets, for example, that are immiscible, and you can create a droplet or a plug. And the trick that we play is that we then immerse this into a fluid that is selective to the solvent that is within the droplet, and it's going to suck that solvent out, a bit like if you left your, say, an apple and you go on holidays, that apple dries and crumples. We're going to do the same thing, but instead of drying in air, we dry it within another solvent. And so this will be familiar to many of you. Microfluidics is very good to create droplets, and they can be very monodispersed, and it's also quite good to impose certain operations, in this case, a drying process. So here we take in this case, a plug. We bring in a fluid that is a selective fluid. It turns these into droplets, and now we're going to extract these droplets. Just to recap what we saw earlier, here you see a number of droplets inside that are clearly, I guess the word we would use is nucleating within this fluid, as well as the fluid shrinking, as well as we found forming a skin, presumably at some point if the process is fast, and eventually solidifying in some kind of directed solidification way. So there's a lot of kind of physical chemistry going on here, and at multiple scales. So clearly from the molecular scale, it must be crossing some kind of phase boundary, and there a glass transition or I guess a gelation process, and eventually some kind of demixing probably as well. So how can we sort of try to break these down into bits so that we can make sense of things? So clearly there is a shrinkage that takes place. So the solvent that is within the drying droplet is being removed, but also we've seen in the case of the levitation, but also in the case here, we might have also non-solvent coming into the droplet. So there's a solvent exchange, a diffusive process at the interface. You might have some kind of demixing, which can be spinodal, for example, or perhaps through some kind of nucleation and growth process, which is the one you saw just on the last image. And then this process is clearly directional, so it's emanating from the interface into the bulk. And so you kind of expect there to be a gradient. And I think a good way to think about this is with this dimensionless number, the Peclet number, that effectively tells us what is the ratio between the speed at which the droplet shrinks, so the velocity of the drying front, compared to the kinetics of the diffusion of the solute. I like to think of this as in terms of maybe a surfer at the beach and a big wave coming, and that the options are, if the surfer is able to swim or to surf ahead of the wave, then the wave will continue to recede and you create a compact particle. But if on the other hand the wave is way too fast, then the surfer will stay at the extremities, and you'll create a skin instead. All the surface will stay out there. No one was able to catch the wave because they couldn't diffuse fast enough. And so clearly at least three processes might be at play. It might be at play, and they clearly will interact with each other. I think Katrina has joined the meeting, so I can show a video of hers that describes this quite nicely. This is a FIB SEM that Katrina took on a flat sheet membrane that has been produced in just the same way, but in a planar geometry through phase inversion. And so the gradient that I mentioned is this. You have a skin that is denser at the top, and then it's kind of more open at the bottom. And in this case, this is a kind of spinodal interconnected structure. So effectively, the same processes that are at play in a planar geometry, for example, when you create membranes through phase inversion, are exactly at play here now in a circular geometry, where you dry radially now, or centrosymmetrically from the extremities of this droplet. So just to take stock of where we are so far, so we say, well, we'll create a polymer solvent droplet. Of course, this polymer eventually is going to become lots of other components that I'll show you in a second. You take a carrier phase, you emulsify this, you immerse them within another selective solvent, and you watch them effectively dry. So you're going to turn a nice droplet, often in dilute solution, into a compact particle. And as you could expect, you get you cross a phase boundary, which is why you see all those droplets nucleating. And in this case here, you might wonder, what's the effect of this phase boundary? What can I do from a thermodynamic point of view that will allow me to control these particles finely? At the simplest level, depending on where you place the phase boundary, you could decide that you'll create a kind of compact particle to some kind of hollow sphere. So this, I think of these as like Maltesers, maybe, because you've created some kind of porosity within, and this some kind of a Ferrero Rocher where the nut has been taken off. But you clearly here you've created a skin. Here you've created a more aerated structure, and of course now instead of having just a polymer, you have another component which could be perhaps an active ingredient. Then you're going to play with that Peclet number of the two components. Think of this. Think of like a fast swimmer compared to a more kind of arthritic swimmer. By designing the way this front recedes around the center of symmetry of this, you can induce a stratification process where you can now have the precise placement of the different materials radially along this sort of drying front. So, I mean, does this work? Is it really that simple? So we take here four solvents, a solvent with an extremely low selectivity, one which is considerably faster, and you realize it's kind of slightly counterintuitive that the one that is the slowest is actually the one that gives you the smallest and the more compact particles. After you say it, it kind of makes sense. You say, well, if I'm going to dry quite fast, then I'm going to get a skin formed quite early. These particles will be large, they will be open, and they will have the ability to contain considerable cargo. And these ones that take the longest time, they are the ones where the swimmers have a lot of time to swim all the way to the core and create a really compact and dense particle. This is kind of a realization of where the particle with the lowest solubility, extracted within this solvent, some kind of ethyl acetate, ends up taking the longest time, but also giving you the most compact particles. Of course, you can take this to the lowest concentration limits and find out what can I do to wrap things around with this technique. And so you can eventually create at such low concentrations shells that are now between hundreds of nanometers to just a few tens of nanometer over here. And so these are really tight skins. The films are continuous. It's as if you are effectively spin coating on a circular, on a spherical geometry. And then, of course, you can contain materials both interacting and non-interacting within these. Now connecting to the molecular scales in the last few minutes of this kind of conversation. What we use for this is scattering. So we can't really do high-resolution electron microscopies, because this process is highly dynamic. We need to arrest the particles, their center of mass. And so we use instead scattering as a non-invasive tool, mainly neutron scattering or X-ray scattering. And we use, again, microfluidics as this potential of allowing you to do these things on the fly, so you can formulate these in real time. And my group has been working quite a lot in developing ways of coupling microfluidic approaches with neutron and X-ray scattering for this discovery of thermodynamics of soft materials, as well as inducing, I guess, these non-equilibrium transitions. So the implementation is quite simple, because these techniques are very, I guess, lenient. You have a microfluidic device. We've spent maybe 3 or 4 years designing the types of cells that you can use, because of course neutrons and X-rays have well, different types of interactions with different materials. We needed this to be transparent, but if you look at this sort of moving up, you can see these droplets are starting to fill up these. This is a kind of special low boron type glass that doesn't really scatter neutrons significantly, so all the signal comes from the sample. And here you see a constellation of droplets, or here, that are moving across our field of view. And you can scan, therefore, spatiotemporally the evolution at the molecular level that these solutions undergo. So the advantage of this is that you can zoom in with as if you do with your smartphone, with your fingers. For example, if you see a region of interest that you locate after first scan, for example, let's say that this is the first set of experiments, you'd like to optimize within this region, you simply reprogram the inputs and you sort of zoom into that region until you find the optimal that you're looking for. In this case, we're looking at the conformation of these excipients within the droplets. And what are the excipients? Well, we've taken as model systems here some silica, some glass beads, if you wish, about 20 nanometers or so in size, some surfactants, because we often need to stabilize these excipients with the help of a surfactant, and a polymer, which is a Gaussian polymer, in this case, in solution. And as we evolve these as a function of concentration or time, we can look at the types of things that scatterers are interested in. So the structure factor, that characterizes the interaction between the spheres within the suspension. And this is important for us to know whether they are interacting favorably with the polymer or not, and whether they are agglomerating and therefore crashing out of solution, or whether they are staying stabilized. So this is a very kind of crowded slide. I don't have time to I guess, to discuss these in detail, but I want to tell you that I think there's a real avenue for us to look at processes in soft materials with neutron and X-ray scattering coupled with microfluidics that we just didn't have before. So we can have this whole succession of steps from the very dilute solution all the way to a concentrated state in a kind of a spatial-temporally resolved way. The same thing to the polymer. So I can come to from the dilute solution and watch, as a function of time, the transformation between the dilute to the concentrated solution, and eventually the formation of the particle. So this is what happens if I have a system that is non-interacting. I've created a thin skin, and here I've suspended a bunch of nanotubes. I've effectively just wrapped these around, a bit like you do in a shrink wrap, for example, for packaging, a bunch of material within a very thin polymer skin. But now let me pick something that interacts. So the same silica particles with the polymer, they have, this is the kind of graph that people in colloidal physical chemistry plot. So this is the concentration of the polymer against the concentration of the nanoparticles. And we come from what's called the gas state, where they are miscible, and we cross into the liquid state where they now demix. And that corresponds to these structures over here, where you can see a stratification. I'm going to select the demixing process that is spinodal, and we'll see kind of what happens here. On the texture of this particle is going to become well, the characteristic of a spinodal. You see that you have a selection of a specific length scale, and you see what looks like a brain-like structure effectively with a well-defined length scale. This is important because it gives us the interconnectivity that we think we need for certain release profiles. So that's the kind of zoom in into that kind of brain-like structure that coarsens over time until it eventually solidifies. And then, I mean, just to tell you that the kind of morphologies that you can access with biphasic systems is endless. So here, for example, you realize that actually you can have quite a lot of say in terms of what structures they select. But I realize that I'm just running out of time. So I wanted to tell you that kind of finished with a claim of success here, which is to say that once we've done this properly, so we've selected a polymer and a colloidal suspension in that kind of spinodal morphology, what you realize is that the release profile is anything but normal. You have these compartments that explode like volcanoes because we've created pockets of high osmotic pressure that once embedded within a good solvent now will explode in this compartmentalized way. You can induce active particles in this way, but significantly the timescales for this are huge and orders of magnitude longer compared to the timescales of the dissolution of just the polymer itself, which is water-soluble polymer that will dissolve within some tens of seconds for these dimensions. And here on the left, you see just some kind of concentration profile that corresponds to a couple of points within this, I think it's over here and here, that correspond to two points of the dissolution of this particle. So I kind of will stop over here. Just remember to thank the people who've done the work and who've helped us. So Rasheen is a student within the CDT in advanced characterization of materials, and she did the levitation work. Will is working on microfluidics, Marco on the scattering, and Christiana worked on these pulsed particles. And most of the funding really was industrial for all of this project. And as I mentioned to you, that nice FIB SEM was from Katrina, and a couple of other people who help us, Lionel in scattering, and Dame Julia also with some aspects of polymer science, and a couple of people who've given us cash. So I'll wrap up over here, and I kind of hope that the talk inspires us to have a chat. So I've dropped my email address right at the start, and so feel free to reach out. I'll be happy to. I'll pass.